Welcome to YouTube live class of Botany. So, in the previous classes, we had started a new chapter, photosynthesis. We shall continue with the next phase of photosynthesis. So, in our previous class, we had discussed about the what is the visible spectrum, what are photosynthetic pigments, and what is the structure of the chloroplast where these pigments are present. So today we shall discuss about the photosystems, absorption spectrum, action spectrum, and the light phase of photosynthesis, where light is harvested and converted into energy of ATP molecules. So recalling the visible spectrum or the photosynthetically active radiation is from between 380 nanometers to 750 nanometers. So this visible spectrum consists of 7 colors, which is commonly called the Vigour colors, and these wavelengths of light are harvested by different photosynthetic pigments. The major photosynthetic pigment being chlorophyll, which absorbs wavelength of light between the blue and the red regions of the visible spectrum. So, how it was known that these pigments absorb different wavelengths of light was by the help of the absorption spectrum, and how efficient they are in photosynthesis was shown by the action spectrum. So, what is the absorption spectrum? So, absorption spectrum is a graph or a curve which shows what is the amount of light of different wavelength absorbed by a particular pigment, say chlorophyll A or chlorophyll B or carotenoid molecules. So absorption spectrum is a graph that shows you the absorption of each wavelength of monochromatic light plotted against its absorbance. So this graph of chlorophyll molecule will show that maximum absorption peak occurs in the blue region and also in the red region of the visible spectrum. Similarly, for carotenoids, the absorption spectrum shows that absorption is mainly in the blue and towards the green region between 500, and so towards the yellow, orange or red wavelengths there is no absorption by the carotenoid pigment. So in the graph we can see in the slide that what is the absorption spectrum and how the action spectrum was given for the chlorophyll molecules. So chlorophyll molecules, so here we can see the absorption spectrum. So the Vigour colors, violet, blue, green, yellow, orange, red. So 400 to 500 is our violet blue region. 500 to 600 is the green yellow region. And from 600 onwards, you have the orange red region of the visible spectrum. So on the other side of the graph, we have light absorbed in percentage. So for chlorophyll A and chlorophyll B, we see two graphs plotted for the amount of absorbance of each wavelength of light. So in chlorophyll A and chlorophyll B, both cases we see there are two peaks that can be seen. The first peak is formed to between 400 to 500 nanometers, and the second peak is formed between 600 to 700 nanometers. So this indicates that the absorbance of light between 400 to 500 nanometers and then from 600 nanometers to 700 is the highest for both chlorophyll A as well as chlorophyll B. The absorbance spectrum between 500 and 600 for chlorophyll A and B is flat. That is, almost no amount of light is absorbed between these two wavelengths, 500 to 600. So that is why the chlorophyll molecules reflect the green light and the leaf appears green in color. So this is the absorption spectrum. What is the utility of the absorption spectrum for photosynthesis? So it has been found that the absorption spectrum nearly corresponds also to the action spectrum. So what is the action spectrum? Action spectrum is a graph that indicates how much of photosynthesis or rate of photosynthesis occurring in each wavelength of light absorbed by a particular pigment. If chlorophyll is the pigment, the light that is absorbed, how much of photosynthesis efficiency can be seen in the particular wavelength of light. So how action spectrum is measured? The action spectrum is measured, or the rate of photosynthesis is measured by the quantum yield. What is the quantum yield? So the byproduct of photosynthesis is glucose, it is water, as well as oxygen that is evolved. So in the action spectrum, it is the amount of oxygen evolved per quantum of light absorbed. So that is the quantum yield that is measured for the action spectrum. So coming to the action spectrum, so this is a graph that shows below the absorption of light and oxygen released, or the rate of photosynthesis or the quantum yield. So take the graph of absorption spectrum for chlorophyll A and chlorophyll B, or in general the chlorophyll molecule. So chlorophyll molecule in the absorption of light, it shows a peak in the blue region as well as in the red region that can be seen in the absorption spectrum. So similarly, correspondingly, we also find that the amount of oxygen released is also showing a peak in the blue region as well as in the red region, where the amount of oxygen released between 500 to 600 nanometers, or in the green region, is almost nil for chlorophyll molecule. So here, this action spectrum also corresponds to the absorption spectrum of chlorophyll molecule. The first action spectrum was given by Engelmann, and he used the algae Cladophora for finding out the action spectrum of chlorophyll molecule. He also used aerotactic bacteria along with the Cladophora algae to find out what is the response of these aerotactic bacteria. So aerotactic bacteria are the bacteria that seek oxygen or move towards the oxygen source. And in his experiment, Engelmann took a test tube, placed a few branches of Cladophora algae in the test tube, and then he exposed the test tube to light which was split by a prism into 7 colors. In the experiment, he found that when photosynthesis started, the aerotactic bacteria were all clumped or started moving towards the cladophora branch, where the branch was exposed to blue light, and also the region where it was exposed to red light. The number of bacteria in the area which was exposed to green light was minimum. So this indicated that in the blue region or the violet region and also in the red region, there was evolution or release of oxygen, and these wavelengths of light were more efficient in carrying out photosynthesis than in the green region. So the accumulation or the abundance of the aerotactic bacteria in these two regions of blue light as well as red light indicated the evolution of oxygen. So this was is known as the action spectrum and the absorption spectrum for each photosynthetic pigment. So the next phenomena is known as what is red drop and Emerson's enhancement effect. So these two are two phenomena which also help to find out the photosystems in the chloroplast or in the higher plants. So first, what is the phenomena of red drop? So this phenomena was proposed by Emerson and Lewis in 1943. So what they did was they used monochromatic light to measure the efficiency of photosynthesis in green algae unicellular chlorella cells. So monochromatic light is individual wavelengths of light, say 650, 670, 660. So each individual monochromatic or a single wavelength of light was used to find out the efficiency of photosynthesis in the chlorella cell. So they also studied the quantum yield, that is the amount of oxygen molecules evolved per quanta of light that was given. So they found that the quantum yield decreased very sharply towards the far red end of the spectrum, that is 680 nanometers and longer wavelength. So when they started for, say, with example 650 nanometers, then 660, 670, and they went on increasing the wavelength, they found that the amount of oxygen that was liberated or the amount of O2 released or quantum yield sharply decreased when the wavelength was increased from 680 nanometers and longer. So this sharp fall in the amount of quantum yield or efficiency of photosynthesis is commonly known as the red drop phenomena in photosynthesis. In the second experiment, they again placed another setup for chlorella cell where they used red light and far red light individually as well as then simultaneously. So in the two setups, in the first setup, the red wavelength of light less than 680 nanometers were given to chlorella cells and the quantum yield was noted. So in again greater than 680 nanometers in the far red light, the amount of quantum yield was measured and noted. So in the third setup, they gave red and far red lights simultaneously to the chlorella cells. So chlorella cells were exposed to red light as well as far red light. That means the wavelengths of light less than 680 nanometer in the red region and greater than 680 nanometers in the far red region. Both of them were given simultaneously to the chlorella cells. So they found that the quantum yield or the rate of photosynthesis was much greater, 2 to 3 times greater than the sum effect when both these wavelengths were given separately. So in the graph also you can see the far red light is on or off, the amount of photosynthesis, the red light area photosynthesis, but when both of them are given simultaneously, the amount of photosynthesis or the quantum yield is much greater than both of them combined together. So this type of enhancement or increase in the rate of photosynthesis was named as Emerson's enhancement effect. The Emerson's enhancement effect gave a better understanding of how the pigments were distributed in the thylakoid membrane of the chloroplast. So how the pigment systems are organized. So later it was found that these pigment systems are collectively called as the photosystems organized in a definite manner in the thylakoid membrane. So for harvesting the light, there is requirement of photosystems which can carry out the process or harvest the light energy. So, what are now photosystems? So, before going to the mechanism of photosynthesis, it is necessary that we understand what are photosystems. So, photosystems are collection of photosynthetic pigments along with different electron carriers and proteins that are present associated with them. So, the types of photosystems, there are two types of photosystems that are present. So the two types of photosystems can be known as photosystem 1 and photosystem 2. and they are named due to their order of discovery as photosystem 1 PS1 and photosystem 2 that is PS2. So coming to the general structure of a photosystem, each photosystem has two specialized chlorophyll molecules of chlorophyll A, which are called the reaction center chlorophyll molecule or the RC. And near the reaction center chlorophyll molecule, you have surrounding them, you have different antenna pigment molecules, which are collection of chlorophyll A, chlorophyll B pigments that absorb different types of wavelengths and they surround the reaction center molecule. So in the antenna pigment molecules, we also have different carotenoid molecules, which we discussed are shield protective molecules that are found outer to the chlorophyll molecule and they form a protective cover. So those are the antenna pigment molecules. In association with the reaction center chlorophyll molecule and the antenna pigment molecules, we also have primary electron acceptor compounds and electron transport system in the membrane to take up the energy that is harvested. So the photosystems here are divided mainly into two types. One is photosystem 1, the other is photosystem 2. Photosystem 1 has chlorophyll A molecules, which is the reaction center molecule, and the reaction center molecule absorbs maximum wavelength of 700 nanometers. So this reaction center molecule of photosystem 1 is called as the P700 molecule, and this photosystem 1 is remaining associated with iron sulfur compound and other electron acceptors. The photosystem 2 has chlorophyll molecules where the reaction center molecule absorbs wavelength of light of 680 nanometer. So this is named as P680. So, and it is also associated with different electron acceptors like pheophytin and quinone. And there are the different antenna molecules. The photosystem 2 also remains associated with the oxygen evolving complex or the OEC responsible for the breakdown of water or photolysis of water. So these are two photosystems that are present in the thylakoid membrane. So what are the two differences between these two photosystems or two pigment systems or photosystem? So photosystem 1 and photosystem 2. The reaction center molecule of chlorophyll molecule is P700 in photosystem 1, whereas it is P680 in the photosystem 2. This photosystem 1 is generally present in the non-appressed part of the thylakoid membrane. That is, the part of the membranes that is directly exposed to the stroma side or is not layered over by another thylakoid. So it is present towards the non-appressed part of the thylakoid membrane where you find photosystem 1. Photosystem 2, however, is present in the oppressed part, that is where one thylakoid membrane is placed over another thylakoid membrane. So there we generally come across the photosystem 2. The photosystem 1 is mainly involved with absorption of longer wavelengths of light greater than 680 nanometer, whereas photosystem 2 absorbs shorter wavelengths of light less than 680 nanometer wavelength of light. So this photosystem 1 and photosystem 2 also show various different type of reactions with in the light reaction. the photosystem 1 is involved in the synthesis of ATP molecule only and does not require any usage of water, or it is not involved in the evolution of oxygen. And whereas photosystem 2 helps in the splitting of water, or it is related with the photolysis of water, and photolysis of water liberates oxygen molecule, so it is involved in the liberation of oxygen molecule. So the photosystem 1 again does not help in the production of NADPH, and NADPH is not formed. So here the NADPH, or the reducing power, also shows a difference of formation in the two pigment systems. So photosystem 1 has iron sulfur compound, and the photosystem 2 is mainly quinone rich and involved in the hydrolysis of water as well as ATP formation. So these are two pigment systems that are found in the thylakoid membrane. So these two pigment systems, along with different electron acceptors and your CF0, CF1 complex or ATP synthase, will carry out the process of light reaction, or the mechanism of photosynthesis will start from the photosystems. So coming to the mechanism of photosynthesis, photosynthesis as a physiological process takes is completed in two phases. One is called the light reaction, or the photochemical phase, that is the light dependent phase of photosynthesis. The other phase is called the light independent phase of photosynthesis, commonly known as the dark phase of photosynthesis. So the dark phase of photosynthesis does not mean it occurs in darkness, but actually it is light independent phase, where the products of light reaction are used in the reduction of carbon dioxide or fixation of carbon dioxide. So coming to light reaction, or the photochemical phase of photosynthesis. First, let us see where the light reactions or photochemical phase occurs. So, light reaction of photosynthesis, or the light phase of photosynthesis, will occur in the thylakoid of the chloroplast. So, the light phase of photosynthesis will involve the two photosystems, that is photosystem 1 and photosystem 2. The two photosystems will be involved along with other carriers. The electron transfer from these photosystems during the light phase is also coupled to the synthesis of ATP, and this is called photophosphorylation. So the major activity that occurs during this photochemical phase is that conversion of photo energy, or light energy, or solar energy into chemical energy of ATP by the process of ATP synthesis. So when ATP is synthesized in presence of light, so it is photophosphorylation phase. So photophosphorylation leading to the synthesis of ATP can be of two types, that is cyclic photophosphorylation and the phenomena of non-cyclic photophosphorylation. What all other activities occur in the photochemical phase is in the photochemical or light reaction. There is also photolysis of water, or splitting of water to liberate oxygen molecule. So here the liberation of oxygen molecule is independent of carbon dioxide fixation during photosynthesis, and also it helps in the formation of reducing power, that is NADPH, or reduced NADPH is synthesized during the photochemical phase. So coming to the two photophosphorylation process and how ATP can be synthesized in this process. The first type of photophosphorylation. So photophosphorylation. Phosphorylation is the phenomena where inorganic phosphate is added to ADP molecule, that is adenosine diphosphate. These two components are negatively charged and they cannot be easily attached to each other, and so it requires the presence of external energy to combine ADP and inorganic phosphate. So here the combination of ADP and inorganic phosphate will be by the utilization of solar energy or light energy. So coming to the phenomena of cyclic photophosphorylation. Cyclic photophosphorylation can be found in photosystem 1. So photosystem 1 is the place where cyclic photophosphorylation takes place, and in this phase there is two molecules of ATP that is produced during the cyclic photophosphorylation. So what happens during cyclic photophosphorylation path is photosystem 1 is first activated by the absorption of light energy. So the reaction center molecule of photosystem 1 is P700. So P700 takes up energy from all other antenna molecules surrounding the reaction center molecule, and then this reaction center molecule gets energy from other carotenoid molecules as well as other chlorophyll A and B molecules present as the antenna molecule, which absorb the wavelength of light less than 700 nanometers. So this energy is transferred to the reaction center molecule by resonance transfer and not by electron transfer. So much of the energy is also lost in the form of heat during the transfer of energy. So when this molecule or the reaction center molecule absorbs energy from the nearby antenna molecule, the reaction center molecule gets photo excited and it loses an electron. So this energy rich electron is expelled from the photosystem 1 or from P700. So when this electron is expelled out, it is first taken up or accepted by the primary electron acceptor molecule, which is a iron sulfur protein. So this iron sulfur protein is the first primary acceptor of electron in the photosystem 1. So from the iron sulfur protein, this electron moves to a ferridoxin molecule which also contains iron, and iron is reduced. So from reduced ferridoxin, this electron flows to a complex of cytochrome B and cytochrome F molecule, and during this flow of electron from ferridoxin to cytochrome, the energy lost from the electron can be utilized in the synthesis of one molecule of ATP. So in the figure you can see ADP plus inorganic phosphate giving you a molecule of ATP, that is seen. Similarly, when the electron flows from cytochrome B6 to cytochrome F, again this molecule or this release of energy also is utilized in the synthesis of ATP molecule. From cytochrome F, the movement of electron is to plastocyanin, a copper containing compound. So here copper is again reduced and plastocyanin then releases back the electron to the photosystem 1 reaction center, which is P700. So this flow of electron here from photosystem 1 is a cyclic path. That is, the electron that was expelled from P700 again flows back to P700 through various electron acceptors. These electron acceptors are arranged in the thylakoid membrane according to their redox potential, and so we find that the electron is flowing downhill to come back to its ground state when it comes to chlorophyll A molecule, which is positively charged or oxidized. So this is the process of cyclic photophosphorylation that takes place during the photophosphorylation path of photosynthesis. The second path or the major path is the non-cyclic photophosphorylation path. The non-cyclic photophosphorylation path is also called as the Z scheme of electron transport, as it resembles the letter Z. So non-cyclic photophosphorylation path also involves different photosystems. That the non-cyclic photophosphorylation path involves both photosystem 1 and photosystem 2. So here the arrow on the left side indicates higher energy level. The molecules remain attached according to their redox potentials, and the photosystem 2 plays a more active role during the Z scheme of electron transport. Here we also will see how NADPH is formed and also how oxygen is liberated during the process. So first coming to the how the electron is transported from the non-cyclic photophosphorylation. So photosystem 2 here is first activated. The photosystem 2 has reaction center molecule of P680 surrounded by the antenna molecules which absorb lower wavelengths of light than 680, and also the shield pigments, the carotenoids. So each of the antenna molecule transfers its energy in a resonance transfer to the nearby molecule, and this energy is ultimately transferred to P680. The photoexcitation process occurs in a femtosecond or in 10 to the power minus 15 seconds. So within a short period, the photoexcitation process takes place. The energy is transferred to the reaction center molecule of P680 and the electrons are expelled. So here you see the movement of two electrons because there are two reaction center chlorophyll A molecule in the photosystems. So here the photo-excited, the P680 molecule is photo-excited or with much energy and loses the electron. So the P680 molecule, after losing the electron, is now positively charged or in a oxidized state. So this molecule that loses the electron is accepted by the primary electron acceptor. The primary electron acceptor here is pheophytin. The first compound that accepts the electron is pheophytin. Pheophytin is a compound similar to chlorophyll without magnesium. Then the pheophytin molecule loses the electron. The electron is flowing downhill, and the next acceptor is plastoquinone. So plastoquinone takes up electron. It gets converted to reduced plastoquinone, and from plastoquinone the electron moves to the complex of cytochrome B and cytochrome F complex. So during cytochrome B F complex movement, there can be synthesis of one molecule of ATP as in the cyclic photophosphorylation phase. So when plastoquinone is reduced at that time and then oxidized, the protons or H plus ions are transported into the lumen of the thylakoid. So H plus concentration will start increasing in the thylakoid lumen. From cytochrome B F complex, the electron passes on to the copper containing compound plastocyanin, which is again further reduced. So plastocyanin, reduced plastocyanin transports the electron to P700. The P700 molecule is photoexcited and it loses the electron. This electron is moving to the next compounds, which is phyloquinone or modified chlorophyll A molecules. From phyloquinone, the electron moves to iron sulfur protein or FES protein, and then to ferredoxin, and then to the flavoprotein enzyme. So in from the flavoprotein enzyme, this electron moves to NADP molecule. So during this flow of electron, we can see the electron that is lost from P680 molecule is not flowing back to photosystem 2. So this electron from P680 has moved and reached NADP plus. So NADP is going to take up this electron. So there is no flowing back of the electron. So what happens to P680, which is now oxidized or it is in a positive state? So the oxidized P680 molecule is a strong oxidant and it will exert a pull on the water molecules that is present in the lumen of the thylakoid. So initially the electron that is lost from P680 creates a hole in P680 or it is positively charged. This positively charged P680 molecule attracts electron from the nearby molecules. So here the role of the oxygen evolving complex comes into action. Water molecule is split into H plus and OH minus ions. These OH minus ions liberate electrons and also oxygen is liberated. The electrons that is lost from water molecule flows one by one to the P680 molecule via the protein tyrosine that is present in the D protein. So here water molecule loses the electron. So water molecule here splits. So this splitting of water molecule in presence of light is called as photolysis of water. So the photolysis of water requires the water splitting complex or the oxygen evolving complex, and the catalyst mainly are manganese ions, chloride ions, and also requirement of calcium ion is seen. So this oxygen evolving complex has a greater concentration of Mn2 plus, Cl minus and Ca2 plus ions, which help in the process of splitting of water. The Mn2 plus ions help in the movement of electrons to P680 molecule. So in this splitting phase of water or the photolysis of water, we find that oxygen molecule is evolved. So the evolution of oxygen is much before than glucose is synthesized by carbon dioxide fixation. So this H plus ions that are released from the splitting of water will move and they will combine with NADP plus and H plus will combine to give you NADPH. So the reducing power that is produced during non-cyclic photophosphorylation is NADPH. So this NADPH has got its hydrogen from the water molecules or splitting of water molecule. So the end products of the non-cyclic photophosphorylation phase are the synthesis of number of ATP molecules is 1 when there is movement of electron from cytochrome B to cytochrome F complex. There is also liberation of oxygen by the splitting of water, and there is synthesis of NADPH molecule which will later move to the Calvin cycle. So the ATP molecules the NADPH molecules will be utilized in the dark phase of photosynthesis where carbon dioxide fixation into glucose takes place. Whereas the oxygen that is formed here is liberated out. So in the entire process of cyclic and non-cyclic photophosphorylation, we find the number of ATP that can be synthesized by the cyclic photophosphorylation process is 2 in number, where the number of ATP that can be synthesized during the Z scheme of electron transport is 1 in number. So a total of 3 ATP molecules can be synthesized by cyclic and non-cyclic photophosphorylation. The number of NADP molecules that are synthesized are 2 molecules in number for the 2 electrons moving. And how is actually ATP molecule synthesized during this process? The movement of electron is coupled with the synthesis of ATP. But the synthesis of ATP is not a direct process as seen in the cyclic and the non-cyclic photophosphorylation. The path of synthesis of ATP forms a takes according to the chemiosmosis method. This chemiosmosis method was proposed by Peter Mitchell in 1961 to explain the process of formation of ATP molecule in the mitochondria as well as in the chloroplast. So, according to this hypothesis or the chemiosmotic hypothesis, the proton gradient or the difference in number of protons or H plus ions across a membrane is necessary for synthesis of ATP by the enzyme ATP synthase. So to know how chemiosmosis occurs, let us have a look at the arrangement of photosystem 1, photosystem 2 and the ATP synthase or CF0 and CF1 unit in the thylakoid membrane. So in the figure, as you can see, one side shows you the thylakoid compartment, the other is the thylakoid membrane, and the lower part is the stroma side. So the stroma side of the chloroplast, the thylakoid compartment or the thylakoid lumen where water is present, and the thylakoid membrane where the two photosystems are present, photosystem 2 and photosystem 1. So when light is absorbed by photosystem 2 and there is expel of electron from P680 molecule, moving of electrons through electron acceptors present in the membrane, P680 molecule is positively charged or it is in an oxidized state. So when it is in an oxidized state, by the help of the water splitting complex, there is splitting of water into H plus and O2 molecules. So these H plus ions are now released in the thylakoid compartment or they are present in the thylakoid lumen. So similarly, when the electron acceptors are again reduced, the H plus ions from the stroma side, for example, your quinone or this quinone, when it gets reduced, takes up H plus ions from the stroma side and then gets reduced. And further, when it is oxidized, it liberates the H plus ions to the thylakoid compartment or towards the lumen side, which can be seen by the black arrow in the figure. So during electron transport chain, again, the H plus ions are released into the thylakoid compartment or into the lumen. So the lumen has a greater concentration of the H plus ions or protons, whereas the stroma side has low H plus ions or proton concentration. So this creates a gradient of proton across the thylakoid membrane. So the thylakoid lumen has a pH of 5 due to presence of more H plus ions, whereas the pH of the stroma side is around 8. So in order to balance this pH of difference of 3, the H plus ions are to be transported to again the stroma side. But the membrane thylakoid membrane is again impermeable to these H plus ions, and these H plus ions are directed through the ATP synthase structure or the CF0 CF1 molecule present again in the membrane. So the CF0 component or the tail part that is embedded and the head part that is CF1 component or the coupling factor is towards the stroma side. So this stroma side contains the head part of the CF1 part which has the ATP synthase enzyme in the inactive state. So when H plus ions are directed towards the from the CF0 component and move through the CF1, their ATP, ADP and inorganic phosphate get combined to form ATP molecule utilizing this proton gradient or the movement of H plus ions across the CF0 and CF1. The head shows conformational changes which activates ATP synthase, helps to combine inorganic phosphate with ADP molecule, forming the ATP molecule. So this is the formation of ATP during the light reaction. So in today's class, we have discussed how light reactions occur, how much of ATP is synthesized, the splitting of water to liberate oxygen, and synthesis of NADPH. So these are the end products of light reaction, which will be later on used during the carbon dioxide fixation of the dark phase. So in the next class, we shall discuss about the dark phase of photosynthesis, the C3 and the C4 cycles of photosynthesis. So thank you all for patiently listening to today's class.